Hi, my name is Anjali Lurie, and today I will be presenting my research on infectious diseases. I focus on assessing potential CWD spillover from cervid species to small rodents. So before I begin, I'd like to introduce you to what prion diseases actually are. So prion diseases are fatal neurodegenerative disorders that are caused by misfolded versions of the protein PRP, which is coded for by exon 3 of the full-length protein gene PRMP. One prion disease that most people are familiar with is bovine spongiform encephalopathy, better known as mad cow disease. As you can guess, that prion disease primarily affects cows. Transmission of prion diseases can occur in several ways, including ingestion of contaminated organic matter, which in itself can lead to cross-species infection, familial inheritance, or spontaneous misfolding of the prion protein. Certain amino acid changes can infer susceptibility or resistance in ungulates. Chronic wasting disease, or CWD for short, is a prion disease that affects deer and elk. We can see here in the lower left how this disease presents itself in elk, causing emaciation in the infected. It is believed that CWD may have occurred spontaneously or been derived from scrapie, a prion disease of sheep and goats. However, scrapie can also affect rodents, as seen here in the bottom left corner of the poster. CWD is particularly widespread, with reports ranging from North America to Sweden to South Korea. It is not entirely clear if or how CWD could spread to non-deer species. However, smaller mammals such as rodents that overlap with cervid populations could be exposed to CWD-like proteins through contact with infected organic matter, such as fecal matter or urine, and act as vectors in spreading the disease. Examining PRMP sequences in rodent species could help to determine this possibility, which in itself would lead to higher prevalence rates of CWD, resulting in a decline of deer populations that could lead to economic losses from reduced levels of big game hunting, as well as potential risk to human health from consumption of infected deer meat. The goal of this study is to determine if the susceptibility variant of the PRMP gene is present in the listed rodent genera in areas where CWD is known to affect deer populations, such as Texas, Oklahoma, and New Mexico. Localities from which samples originated are shown in Figure 1. In doing this, we examine the possibility of cross-species infection from deer to rodents. To accomplish this goal, we obtained tissue samples of the genera from Texas, New Mexico, and Oklahoma from the Natural Science and Research Laboratory. We then extracted genomic DNA using a DNeasy blood and tissue kit. The PRMP gene was amplified via PCR, purified, and cycle sequenced. The purified PCR amplicons were sent off for sequencing. Those sequences were then viewed and annotated using a program called Sequenger. This data was combined with sequences of other rodents obtained from GenBank and used to construct a phylogenetic tree using another program called Mr. Bayes, with parameters set at 10 million generations, burn-in of 25%, sampling frequency every 1,000 trees, and 50% consensus rule, which is shown in Figure 2. We have just received these sequences back this week, so in the coming weeks, we will visualize and annotate those sequences using Sequencer to determine if these species have the key amino acid changes of the susceptibility variant of the PRMP gene. So far, we have successfully generated sequences of PRMP and compared those to other species within order Rodentia, and built a phylogenetic tree, which is shown here in Figure 2. This gene appears to follow the accepted rodent phylogeny, with most nodes displaying a high level of certainty. As you can see here, this node has a 1, which represents 100% certainty. So at this node, we are 100% confident that this branching pattern is correct. I've indicated the 6 genera for which we have produced sequences, and all of which follow the expected phylogenetic relationships. As you can see, Ectotomies and Sciurus fall into one monophyletic group, which is expected and represents family Sciuridae, the squirrel family. Dipotomies and Ketodipus over here fall out into the same monophyletic group, which is expected and it represents family Heteromyidae, which is other mice. And lastly, We have Peromyscus and Neotoma, which fall into family Cricididae, New World mice and rats. In each case, we saw the we saw what was expected in rodent phylogeny. If we do detect this susceptibility variant, it could have important implications for wildlife infectious diseases as well as human health, as it could suggest rodents acting as vectors and spreading CWD, leading to higher prevalence rates and a possible decline in populations. In turn, this decline could lead to economic losses from reduced levels of big game hunting and potentially human health risks from consumption of infected deer meat. So before I end my presentation, I'd like to thank the Honors Undergraduate Research Scholars Program supported by the C.H. and Helen Jones Foundations. Many thanks to H. Garner and K. McDonald of the Natural Science and Research Laboratory for providing these tissue samples. And of course, thank you so much for viewing my presentation. If you have any questions, please reach out to me. I would love to hear them. Thank you.